the course, as I understood it, is mostly for single-cell RNA sequencing analysis. But there's a lot of other applications that are becoming more common in the single-cell field. And one of them is the single-cell attack sequencing. So basically, that's an assay for looking at accessible chromatin. And this, in this illustration here to the left, you can see, that the DNA is compacted into the chromatin with the help of the nucleosomes. So the DNA is wrapped around the nucleosomes and the histone proteins. And then, of course, the open chromatin is more available for transcription factors. So when you introduce the TN5 transposase that you also use in segmenting the library for the RNA sequencing methods, the open chromatin is more accessible to the TN5 enzyme. Thus, it will cut and tag those open chromatin fragments or open chromatin and fragment them, and then you build libraries from these fragments and you sequence and you map back to the genome. So basically, in the end, it's a proxy for regions in the DNA that are open for regulation. So it's data that could be added to the RNA sequencing data. And we have run a lot of projects doing only single-cell attack sequencing data. But now there are methods available where you can do both the RNA-seq and the single-cell attack-seq readouts in the same cells. So I think we will see more of that in this coming year. So I mentioned combining single-cell attack-seq with RNA-seq, and then combining a protein readout with single-cell RNA-seq. But there are many of these multimodal omics methods around. But I think they are very specific for your question. But we also have tested now running single-cell cut and tag. I don't know if you know about that method, but it's in somewhat similar to single-cell attack-seq. But while in single-cell attack-seq, you just cut in open chromatin, the transposase cuts in open chromatin. But with single-cell cut and tag, you can look specifically at transcription factors or histone marks with an antibody. So then you have an antibody to the transcription factor of interest or the histone modification of interest, and then you cut there. So then you get to know if your transcription factor is at the situation of is at the location of is there to regulate the gene expression. Well, that's what I want to say.